Hello, welcome to Gurukul's CAT 100 series, a series that's designed specifically to take you from your study room to the corporate boardroom. I am Prady Sharma and this is Verbal Mastery with Prady. Hello guys, and welcome back to another video on the 100 Days to CAT series of Verbal Mastery with Prady by the Gurukul. Now, in case you want to take this up as a test, this passage as a test, then all you've got to do is go on to www.gurukul.org.in and register for the 100 Days to CAT test series. Obviously, this is a free series for all of you people. Alright, all you've got to do is just register on the above thing and you'll get 100 tests of quant, LRDI and verbal ability for free. Right, now in case you don't want to take it as a test and you want to follow along with Prady on this video, then all you've got to do is pause when I ask you to pause, measure your time, answer the questions and then look back for the questions and passage explanations. So without any further ado, let's get into it. Okay, so this is your first paragraph. Now pause the video here, read this and then we'll move on to the second one. Alright, so this is your second paragraph. Again, pause the video here, read this paragraph and then we'll move on to the third one. Now this is your third paragraph. Pause the video here, read it and then we'll move on to the questions. Okay, your question number one. Put on your timer, just get to know how much time you're taking to solve each question. That's very important for CAT, by the way. So pause the video, answer the question and then we'll move on to the second question. Okay, question number two. Again, pause the video here, answer this question, then we'll move on to the third question. Alright, question number three. Pause the video here, check your time and then we'll move on to the last question of this passage. Okay, question number four. Pause the video here, answer the question. Right, now that you've done answering all the questions, put those answers down in the comment box. Once we finish the passage explanation and then we go into the question explanations, we can check how many of you have got them all correct. Alright, so let's get into the passage explanations. Okay, so what is he saying here? He's saying that genome editing, also called as gene editing, is a group of technologies that give scientists the ability to change an organism's DNA. So what I'm going to do from now is I'm going to underline all the important parts and then I'm going to look through it at the end again, okay? So we're talking about genome editing, okay, and it's what is this? It's the ability to change an organism's DNA. So we can change a person's DNA using this genome editing. These technologies allow genetic material to be added, removed, altered or modified at a particular locations in the genome. Okay, that is nothing but the ability to change, right? So it's not a new idea for it, it's the same idea being repeated again. Chalo. Several approaches to genome editing have been developed. Okay, great, same thing again he's saying, but he's saying that a lot of approaches have been developed. That's great. A recent one is known as CRISPR-Cas9. So now he's getting into the specifics. So first he started off with genome editing. Okay, then he told us about the definition. He said that there were a lot of approaches and now in the approaches he's talking about CRISPR-Cas9. That's one approach. Right, so which is short for whatever, clustered, regularly, interspaced, short, palindromic repeats and CRISPR associated protein 9. Okay, doesn't matter what it's short for. We'll try and understand what it is later. Okay, so the CRISPR-Cas9 system has generated a lot of excitement in the scientific community because it's faster, cheaper, more accurate and more efficient than any other existing genome method. So what do I understand? In short, I understand that this has taken Horlicks. Okay, so this is faster, shorter, what do you call, cheaper, right, accurate. So this is basically the that guy in that advertisement who has taken Horlicks, right? Okay, so CRISPR-Cas9 was adopted from a naturally occurring genome editing system in bacteria. Great, now I understand that this has come from the natural process of bacteria. So the bacteria captures snippets of DNA from invading viruses and use them to create DNA segments known as CRISPR arrays. So what is happening here? This is the major idea that we're getting from the passage. The bacteria is trying to capture snippets, that means small parts of DNA from invading viruses. So the viruses that are invading it or the viruses that are attacking it. okay, and use them to create DNA segments known as CRISPR arrays, and that those small parts which have been taken from these viruses are being again used to make new DNA segments. That CRISPR array allows the bacteria to remember the viruses, okay, or closely related ones. If the viruses attack again, the bacteria produces RNA segments from the CRISPR arrays to target the virus DNA. Okay, so now see what's happening. Now, virus is attacking bacteria. Okay, so this virus, let's say, has some kind of a DNA, and I'm breaking this part and I'm storing this. The bacteria is storing this. Okay, this is known as its CRISPR array. Now, when this virus again attacks, okay, when this virus attacks again, then this part which is there, right? So this part, it remembers, the bacteria remembers this. Okay, then what happens? It generates its RNA. Okay, so that is what's happening. Yes, so the bacteria then uses a Cas9 or a similar enzyme to cut the DNA apart, which disables the virus. So it generates an RNA, and then this gives us Cas9, which cuts this part, okay, which cuts this part so that this virus is disabled. So I hope you guys are understanding what's happening. This is a little technical, but it's easy to understand if you are looking at the diagram overall, right? Let's go to the second part. So the CRISPR-Cas9 system works similarly in the lab. So just like how it works in bacteria, it works in the lab as well. Researchers create a small piece of RNA with a short guide, so that this part that we've taken from the, virus, right? So this part we call as the guide over here. Okay, yes, guide sequence that attach, okay, to a specific target sequence of DNA in a genome. So what's happening? This guide sequence is getting attached to a specific target sequence of a genome in a DNA. So it's getting attached to this part like this. Okay. The RNA also binds to the Cas9 enzyme as in bacteria. The modified RNA is used to recognize the DNA sequence, and the Cas9 enzymes cuts the DNA at the targeted location. So again, what's happening is the same thing. Same process is being repeated even in humans, even in the lab. That's what he's talking about. So although Cas9 is the enzyme that's used most often, other enzymes are also can be used. Okay. So Cas9 is most likely to be used, but there are other enzymes also that you can use. Okay. So here we don't get any new idea. Okay. So what we understand from this part till here is that bacteria and lab both are same in terms of CRISPR-Cas9. Okay, so that's all I understand from here. Now, once the DNA is cut, researchers use the cell's own DNA to repair machinery, or to add deleted pieces of genetic material, or to make changes. Okay, so now whatever is happening, once that DNA has been cut, okay, so researchers are using the cell's own DNA over here to make some kind of a changes. Okay, now comes the point of genome editing. Genome editing is of the greatest interest in the prevention and treatment of human diseases. Wow, great. So now what do I understand? I understand that genome editing is very, very important for us to prevent and treat human diseases. Right, human diseases. Great. Okay. Currently, most research on genome editing is done to understand understand diseases using cells and animal models. So right now we are using it on animal models. We have not done a lot on humans already, but yes, scientists are still working to determine whether this approach is safe and effective for use in people. Right. So we have not done much on the humans. Why? Because we are trying to see if this actually is safe or not for humans. Okay. It's being explored in research on a wide variety of diseases, including single cell gene disorders such as cystic fibrosis, hemophilia, and sickle cell disease. Again, these are examples. Okay. So right now, what do I understand more about genome editing? Okay, done in animals. Okay, it's done in animals. Checking for humans. That's all I'm going to write over here about my genome editing. Right? It also holds promise for treatment and prevention of more complex diseases such as cancer, heart disease, mental illness, and human immunodeficiency virus, which is a HIV infection. So it can potentially cure a lot of diseases, is what he is trying to tell us. Great. Okay, let's move forward. Ethical concerns arise. So now another shift comes in the idea. There's a new shift in the passage. Okay, so ethical concerns arise when genome editing is using technologies such as CRISPR-Cas9 is used to alter human genomes. Okay, so when is this ethical concern coming? Ethical concerns are arising when we are trying to alter human genomes. Okay. Right, so most of the changes introduced with genome editing are limited to somatic cells, which are cells other than the egg and the sperm. Okay, so they have nothing to do with fertility. So most of these are related to somatic cells. Let's just remember this for now. Okay, these changes affect only certain tissues. Okay, so he has clearly said only certain tissues and are not passed from one generation to the next. So they cannot be inherited. That is another major idea that we have to remember. However, changes made to genes in egg or sperm cells, which are known as the germline cells, or in the genes of an embryo, could be passed to future generations. Okay, so if you're doing it for somatic, so what do we understand from here? So the ethical concern is about changing human genomes, right? So currently whatever research is happening, it's for somatic cells, right? So somatic cells cannot be inherited, I can say this, right? So here it is okay, but when we are talking about germline cells or embryo, it is not okay, right? Okay. Now let's look at this. Germline cells and embryo genome editing can bring up a number of ethical concerns, okay, including whether it would be permissible to use this technology to enhance normal human traits. So why are we not trying to do this? Because is it permissible or not to enhance human traits? That is what we are trying to see. Okay, based on concerns about ethics and safety, germline and embryo genome editing are currently illegal. Okay, so we don't want people to change the sperm and egg cells, and we don't want them to mess around with the embryo a lot. Why? Because it's not really safe. We don't know if it's safe yet or not, and there are a lot of ethical concerns with it. So these are all the ideas that we get from this passage. Now quickly let's get into the questions, because we're done with the passage explanations. I hope you understood. Please let me know in the comments if you need me to explain it further. If there are any doubts, please don't hesitate to put it down in the comment box. Let's go into the questions. Chalo. Question number 1. According to the passage, all of the following are true for CRISPR except. So what is not true for CRISPR? He's just saying that there are 3 statements which will be true for CRISPR. What's that one statement that's not true for CRISPR? Right. So it has the potential to prevent and cure diseases. Obviously this is true for CRISPR. Right. It's capable of altering somatic cells but not germline cells. See, again, it is capable of altering. We are not yet sure if we should or not, but it can. So this is also true. But not germline cells, sorry. So it can alter germline cells also, right? So since he said not, this becomes false. It plays a role in regulating bacterial immunity, obviously, otherwise we wouldn't have gotten to know about CRISPR at all, right? Obviously has a role in the bacterial immunity, so this becomes true again. Its promise also raises ethical concern. Yes, just because of the number of things that it can do, we are facing ethical concerns about it, right? So this also becomes true. So what is the only thing that's false? Obviously, this B option is the only option that is false. So our correct answer should be B. Now let's go to the second question. According to the passage, which of the following is true of Cas9? Okay, so what is true of Cas9? What does Cas9 really do? The enzyme Cas9 activates the cell DNA to repair mechanisms. Okay, I'm going to stop reading right there, because first things first, Cas9 does not activate cell's DNA, right? From the passage, we've never heard that Cas9 activates any cell DNA, right? So this will be wrong. The target location where Cas9 cuts the DNA cannot be predetermined. If it cannot be predetermined, how are we saying that it'll get attached somewhere and then it'll go and cut it? So obviously this is also wrong. Cas9 is an enzyme in bacteria, great, okay, that cuts foreign DNA. Yes, that is what we've learned, right? It cuts the DNA of the virus. Yes, so this could potentially be our answer if something else does not show up to be more promising. Cas9 is an enzyme in bacteria that cuts native DNA. Native DNA means its own DNA. It does not cut its own DNA, right? It cuts the virus DNA, right? So our correct answer should be C. Okay, let's go to the third question. Primary purpose of the passage is to, so what is the passage really trying to tell you? That's what is the whole question. Highlight the ethical concerns that arise out of tampering with genomes. Great, the passage is telling you this, but my dear friends, this is just a partial scope choice. Out of the 3 pages of passage that we've read, this is only present in the last page. Just the partial scope, okay, not the complete scope of the passage. Chalo, explain the, so this should not be our answer. Let's just get that straight. Explain the findings of research conducted in the area of CRISPR-Cas9 technology. Great, it is again talking to me about what is happening, but this time it's kind of too broad. Okay, it's not really very specific as to what the passage is really telling. It's just saying explain the findings of research. Now, it's not only the findings of research that he's talking about, right? So I'm gonna remove this as well. Examine how CRISPR-Cas9 technology has the potential to affect the future of humanity. Oh my God, wow. Again, extremely broad, right? So he's talking about the potential to affect the humanity, and that's not the only thing. We are talking about the ethical research, which also come into this, right? But we're not really looking at the findings and all over here, so we can take this off. Next, explain the CRISPR-Cas9 approach to genome editing and the ethical concerns. So yes, here we are talking about the CRISPR-Cas9 approach and we are talking about the ethical concerns in genetic modification. Both of these fit. So the correct answer should be D. Now let's go to the last question very quickly, okay? So till now, how many did you get correct? I hope you got all the 3 correct, right? So, which of the following statements can be inferred about the ethical concerns? So, what can I understand about the ethical concerns as he's told in the passage? That's what's the question about. The primary ethical debate is whether genome editing should be used for treating diseases or only for enhancing human traits. See, there's no question like that. Okay, in the passage he's never said it's either this or this. He's actually concerned about enhancing human traits. Okay, so this clearly should not be my answer. Most countries currently prohibit all forms of genome editing due to, oh my God. So guys, all forms of genome editing? No, not really. They're only telling that don't do germline or something, right? Yeah, germline kind of editing. This is what they're trying to say, right? So this is something that we call as an extreme answer, and we can remove this as well. Ethical issues become particularly pronounced when genome or genetic alterations have the potential to be inherited. Yes, so that germline thing, the embryo thing, that's all about the inheritance, right? So this is yeah, so this is something that I can actually understand. This is something that can be my potential answer if nothing else better shows up. Okay, so altering somatic cells raises fewer ethical concern than altering germline cells because somatic cells changes are more easily reversible. Great. Till this part, to be honest, it's actually better than here. But the problem is, because somatic cell changes are more easily reversible. Where did he tell us that this is more reversible than that? Nowhere did he talk to us about reversibility at all, right? So obviously I cannot take this as my answer. My correct answer should be C. Right? So with this, we're done with all the 4 questions. Tell me in the comments how many questions you've got correct and why you got those questions correct. If there's anything that you've gone wrong in, please try to analyze why you've gone wrong. And if you cannot, then please leave that down in the comments so that I'll tell you why you went wrong. Just give me your thought process in the comments and we'll meet in the next video.
Hope you enjoyed learning from today's session.
This series is designed to help you with your CAT preparation and practice daily. We are with you in this journey.
To watch the next video, click here. And to watch Gurukul's CAT 100 series, study room to boardroom, the entire playlist, click here.
If you learned from today's session, like, share, and subscribe.